your biochem recap, tell me, what does biochem stand for? Biochemistry, what does that stand for? What does it stand for? Bio means life, and chemistry means reactions. So biochem is the reactions of life, right? So if you got this category majority wrong, I would star it, review it. So biochem, our unit 1 started week 1 of school with our lovely water molecules. What do you know about water? What do you know? You can dissolve in it. Okay, what do you know about the chemistry of water? It's H2O. And water is always. Here, let me draw my water molecule. What am I missing on it? What do I not have on it? Huh? Is this a good water molecule? No, what is it missing? Not the bonds. It has bonds. Water is what? There's the Nope. Chemistry. So to the P. Polar. So what do I need to add to this picture? Negative and positive. Right? Water looks like that. Because water looks like that, it's going to form what? What does it do? Hydrogen bonds, right? So it goes to another water molecule. And it says, hey, I like your electrons. Can I have some, please? And it's going to form our lovely hydrogen bonds. Are hydrogen bonds weak bonds or strong bonds? Weak or strong? Weak, right? They're very easy to break. We're constantly making and forming them. And because our hydrogen bonds are weak bonds and they form between water, it gives water all its unique properties that help it support metabolism in the body. So our first property water gets is adhesion. What does it mean to adhere? Sticks. Who do I stick to? Other water molecules. Nope. Adhesion. I stick to others, not other water, just others. Okay. What does it mean to be cohesion? Cohesive. What did you say earlier? Water to water. Okay. What's capillary action? Up the stem, roots to leaves, right? Roots to. What's surface tension? They stick together, stick and form a. There's close. Looking for a different word. Another B word. Not bond. Not a barrier. Think about the pennies. Bubble. Coadhesion form a bubble. likes the bubbles? Who likes when it forms a bubble? Specific organism. Bugs. They love to bring up about the bugs loving water for their metabolic activities. High specific heat means it does what? Tolerates high heat. It's able to keep it the ocean water, right? It can retain heat. And universal solvent means it can do what? Dissolve. I'm looking for the word dissolve. Breakdown's fine too. Able to dissolve for chemical reactions. It's great at giving us nutrients. So that was all week 1. Water. We did the drops in the penny. We did the plants with the food color for capillary action. I think I showed you the walking water with the cups. That was all week 1. Week 2 was focusing on macromolecules. So our biomolecules are all made up of 6 elements that make us living. What are the 6 elements of life? Carbon, oxygen, phosphorus, nitrogen, 2 more. 2 more. You're missing one big one. Sulfur. That's not the one I was looking for. What's the second one? Hydrogen, right? Chomps. Remember we talked about elements, chomp? You can't see that. Chomps. Carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. Our first biomolecule is carbohydrates. What do carbohydrates look like? Hexagons, right? They're very characteristically, typically a hexagon. Joined together, whatever. What are the 3 elements in carbohydrates? What are the 3 elements? Carbon and hydrogen and oxygen, right? Chow. What is the monomer? Monosaccharide. What does mono mean? What's saccharide mean? Sugar. What's your example? Glucose. Because os means sugar. What's a polymer? What's a big carbohydrate? A polysaccharide, but what, give me an example of one. Give me an example of a polysaccharide. You probably ate this for breakfast. Huh? Bagels. Bagels are made up of bread, which is made up of, starts with an S. Starch. Starch. Function. Purpose. Short-term energy. They also make up what? Cell, not membranes. Cell walls. Where do I make these? They love to ask me that. Where do I make carbs? How do I make carbs? What process do I make carbs in? Photosynthesis, which happens where? Chloroplast. Okay. Lipids. Lipids look like what? What do lipids look like? Yeah, they have the little tails, right? The thing, the squiggly lines. What are the elements in a lipid? Carbon, hydrogen, and oxygen. And how much oxygen do they have? A few, right? A little. Monomer. A little bit harder. One starts with a F. Fatty acid. And one starts with a G. Glycerol. Glycerol. Right, that's what we consider the monomers. What are an example? What's an example? Fat. What else? What's fat as liquid form? Oil. What's fat in a solid form? Wax. And you told me this one is the what part? Cell membrane. Function. Long-term energy. What else? What did you just tell me it's from? Cell membrane. Why do you store fat? Warm. Because it's a insulator. And one more thing with lipids, people take these to like get super ripped. Steroids. What are the steroids? You talk about a guy taking steroids. Why? What is that? They're just pumping their body full of which one? Testosterone. That's what steroids are. It's just testosterone or estradiol, your sex hormones. Proteins look like what? Oh, wait, let's go back. Lipids. Who makes a lipid? Where are lipids found? What organelle? Starts with an S. 3 words. What's on your head? Smooth. So the tip of your head. Smooth ER. Good. Proteins. What do proteins look like? They do, but what do they look like? A tube. Okay, I see. I see what you're doing. I see what you're doing. The tube, the channel. Okay. Normally they're gonna be a bead, like a beaded necklace. They're like a necklace. We see them as those channels because they have like coiled up, but they kind of look more like a necklace. What elements are in them? Hydrogen, nitrogen, right? The appearance of N tells me it's a protein. What's their monomer? Amino acid. Example. Enzymes. Give me a example of an enzyme. You probably use this enzyme because you can't break down milk. Lactase. Give me another one. Work in the gym, trying to build something. Muscles. There's some proteins in your hair. You might go to the salon and get a treatment for this. They put this in your shampoo. So to the K. Keratin. Dandruff was a good one, though. So what's the purpose? Function. What do I use them for? Dylan, you said something earlier. Enzyme. Yeah, I'm going to say cell structures. Because that'll cover all the stuff for growth. And then all the transport stuff, too. Something similar to lipids as well that we talked about earlier. They're the rest of your hormones. So testosterone, estradiol is a steroid. Everything else, your cortisol, right? Hormone. That's protein. And they also do your immune system. Okay. Nucleic acids at the bottom. What do they look like? What do they look like? You only got two of them. What do they look like? That's base pairs. What's the overall structure? What do they look like? What does DNA RNA look like? One single stranded. Okay, but what's that shape called? Helix. Dylan's really quiet. What are the elements in nucleic acids? All of them. The presence of N and P tell me it is a nucleic acid. Oh, I almost forgot. You will get some S's popping up. What about a monomer? Nucleotide. You said our examples are DNA, RNA. What's the function? Makes up DNA, which is what does what? What is DNA? Genetic information. Yep. And then it does one more thing. What does RNA do? What's RNA? RNA does what? Makes What do you say, amino acids? Yeah, good. Makes protein. Protein synthesis. And that was week 2 of school. Week 3 kept going, and we continued on with our enzymes. You just said enzymes are what macromolecule? Proteins. Enzymes are proteins. I have another pen color. So for my enzymes, I get this lovely little reaction chart that always pops up for us. What does this label does? Oh, can't see my finger. What's this? That's the enzyme, yeah. And enzymes bind to they're at the active site, but they bind to the what? Substrate. What do you get at the end? Product. Do you remember this little graph thing? Not graph, graphic. I'm giving it here. So enzymes, what do they serve as? What's their official title? They are a catalyst. And what do catalysts do? Speed up reactions. By doing what? What do they do? Lowering activation energy. So if I have lower energy, right, a lower hump, a lower start, what happens to my reaction? My reaction will go faster. All of my enzymes end where? What are the endings? What's their suffix? Ase. Okay. So when I do my enzymatic reaction, does my enzyme change? Or is it the exact same? Exact same. I can reuse it, right? Recycled. So if I had a little reaction A plus B yields AB, my enzyme only gets applied to this little arrow sign, to the progression of the reaction. And then we love to affect activity rates. They can get affected by what? pH or temperature or last one. So to see concentration. Temperature and pH can cause my enzymes to do what? Denature, right? That was your vocab word of the unit. Your lovely little graphs. It shows you that enzymes love a optimal pH. They're best Anything too low, they're not doing too hot, they're probably going to denature. Anything too high, they're probably going to denature. In terms of temperature, they have their best temperature. Anything too high, they start breaking down. Too low, they're not doing too hot. With temperature, they only typically denature at low temperatures. They just get very slow at low temperatures. They denature at high temperatures. Concentration can affect how much I can use. If my substrate, if I have a lot of substrate, eventually I'm going to run out of enzymes. All my active sites get saturated. Remember that was when we were chopping the toothpicks, right? You only had 2 hands to break the toothpicks. You had 1,000 toothpicks. You're not breaking it very fast. Eventually you're going to reach your fastest point. You can also denature it, not denature, also affect the activity rate based on your concentration of enzyme. If I had all of you try to break the toothpick with only 4 toothpicks to break, we can't go any faster once everyone has one. You're gonna run out of substrate. There's no more substrate available. And that was week 3. I did miss something, so I'm gonna go back real quick. For proteins, what do I make proteins? What organelle do I make them in? What is it? Not the ER. Close. Ribosome. I must have missed that part. And then where do I make nucleic acids? What organelle do nucleic acids live in? Nucleus. Yeah, I was going to say you better know that one. Good. Just to make sure I was complete. And that is the end for Unit 1. Good work, guys.